In the 1930s, two doctors, Simon van Creveld and Richard Ellis, met on a train. Remarkably, each had encountered a family with similar developmental problems in their bones, teeth, fingers, limbs, and hearts. A decade after Ellis and van Creveld published their findings, a doctor named Helmut Weyers observed a similar developmental disease, though this disease was inherited in an autosomal dominant fashion. In the 1990s, Dr. Claire Francomano tracked down families in which Ellis-Van Creveld disease was repeatedly inherited and mapped the responsible locus to a point on the fourth chromosome. During the first decade of the new millennium, Doctors Judith Goodship and Victor Ruiz Perez identified EVC and EVC2 as the mutation-carrying genes relevant for Ellis-Van Creveld disease, and EVC2 as the gene relevant for Weyers disease. Goodship's team clearly demonstrated that the proteins encoded by EVC and EVC2 concentrate at the base of cilia and are a critical part of the Hedgehog pathway, coordinating the development of bones, teeth, fingers, limbs, and hearts. In this issue of Developmental Cell, our team identifies FCAB7 and IQCE as EVC2 interacting proteins. We define the conserved genetic domains responsible for these protein-protein interactions and demonstrate them in vitro. Our sequence analysis suggests the molecular basis for the cellular phenotype seen in Weyers disease. We test our hypothesis by genetically removing FCAB7 in cultured cells, and we find the interactions between FCAB7 and EVC2 is the prime determinant of the cellular phenotype seen in Weyers disease. Most importantly, we demonstrate that IQCE and FCAB7 are critical for the intercellular interpretations of the signal from the Hedgehog ligand. We see that the Hedgehog signal splits at the EVC-EVC2 complex, a rare biochemical observation supported by the tissue-specific effects seen in patients with EVC and EVC2 mutations.